In this video, I'm going to draw the circuitry of the cerebellum, and I want you to draw along with me following this template that has 3 layers of cerebellar cortex. So the outermost layer of cortex, so this right here is cerebellar cortex. And unlike the cerebral cortex, the cerebellar cortex has 3 layers. And they are 1, 2, and 3. And then this is the cerebellar white matter. And deep to the white matter, there are the deep cerebellar nuclei, which don't eat greasy food, which stands for the dentate, that's the D, and it's sort of worm-shaped. There's emboliform, which is the E. There is globose, and these are called the interposed nuclei, interposed because they're next to each other. And then the last one is vestigial, the F. So don't eat greasy food are the dentate, emboliform, globose, and vestigial nuclei. Now, what we're going to do is to add the cells in the cerebellum. So we'll start with the only cells that project from the cerebellar cortex out. And these are the Purkinje cells. They have a cell bodies that are lined up in a nice, beautiful row in this second layer of cortex, which is named appropriately the Purkinje cell layer. And the Purkinjes are GABAergic. They are inhibitory with GABA onto the deep cerebellar nuclei. So I'm going to draw them. They have this beautiful dendritic arbor, which you've probably seen in beautiful art, and like neuroscience art typically shows the Purkinje cells because they have these massive tree-like dendritic arbors. All right, let's zoom out a little bit so you can see this. So again, the Purkinje cells they have their cell bodies in their Purkinje cell layer. They have their dendrites in this layer above, called the molecular layer. And their axons come down here into the granule cell layer. So the Purkinjes are the main output neuron of the cerebellum. They send axons all the way down here to the deep cerebellar nuclei, which are the dentate, interposed nuclei, the emboliform and globose, and the vestigial. Now, the Purkinje cells receive inputs from 2 main sources. The mossy fiber nuclei are coming from lots of different places. It's the pontine nuclei, Clark's column, external cruneate, the spinal border cells, vestibular nuclei. Some of these we've learned, some of these we haven't, but essentially you're taking information from spinocerebellar tracts, pontocerebellar tracts, and vestibulocerebellar tracts. And these are all coming in, and they are going to synapse on cells in the granule. So I'm going to just make multiples here. These are synapsing on granule cells. Now, what are granule cells? Granule cells are cells that make up the granule cell layer, and I'm going to draw them here in red. Granule cells are incredibly dense and make up a huge majority of the cell counts in both the cerebellum and the central nervous system as a whole. They have axons that come up into the molecular layer. The axon splits into parallel fibers. These parallel fibers are excitatory onto Purkinje cells. So everywhere along the parallel fiber, you'll get a synapse where that parallel fiber will excite the Purkinjes. So again, these are the granule cells that end in parallel fibers. And these, in green, are the mossy fibers, coming from the vestibular nuclei, coming from the pons, and coming from the spinal cord. And these are also excitatory onto the granule cells. And on their way in, they will send excitation onto deep cerebellar nuclei. Now, why would you do that? Well, the fast route is to excite the deep cerebellar nuclei. The long route is to excite the granule cells. The granules excite the parallel fibers, And the parallel fibers, then you're getting an excitation of the Purkinje cell. But think about it, the Purkinje cell is inhibitory. So you're turning the deep cerebellar nuclei on with this branch, and then through this longer synapse, you're exciting the Purkinje cell, which is inhibitory, which is then the off signal. So you're doing a very quick on-off through this unique loop again. The other major input into the cerebellum, and I'm going to draw it over here on this side, is the inferior, let's not put it there, let's put it all the way out here because, we're talking about something that's in the medulla of the brainstem. The inferior olive, which is another squiggly nucleus, not to be confused with the dentate of the cerebellum. This is in the medulla oblongata. We have the inferior olive nucleus. And the inferior olive nucleus sends what are called climbing fibers all the way up. So these are climbing fibers. And they come in and they do just what it sounds like. They climb the branches of the Purkinje dendrites, and all along the way they excite the Purkinje cells. So they, again, are an excitatory neuron. Guess what? They also do the same thing where they send a collateral branch to excite the deep cerebellar nuclei. So what happens? You get an input, actually it's coming from the red nucleus. You can think about the red inside of an olive. Comes down to the medulla, synapses in the inferior olive. On the way in, the first stop is, let's turn on those deep cerebellar nuclei. And then the longer route is to climb the tree of the Purkinje cell dendrite, excite all along the way. Remember, Purkinje cells are inhibitory, so you're exciting an inhibitory neuron, which means you're sending first an on signal through this pathway, and then right away you turn an off signal. Again, notice that we're shaping movement by a very short on, and then shaving it away with an off. In general, the mossy fibers tend to be sort of reporting back what happened. So vestibular information is what's the head position. Spinal cord information is what's the body position, like proprioceptive information. And then we have these pontine nucleus. What were the plans that were made for that movement? Whereas the climbing fibers send us error signal. Like, this is what we planned to do and this is what happened. There was an error and I'm going to fix it, is the climbing fiber. That's a little bit simplified, but that's one way you can think about it. There are multiple GABAergic interneurons. Now, the story gets much more complicated. And I'm going to draw them in pink. I don't want you to worry too much about the Golgi cells, the basket cells, and the stellate cells, except to know that they are inhibitory interneurons of the cerebellum that can change the circuitry and modify. Again, remember, the cerebellum is not a structure that makes movement. The cerebellum is a structure that refines movement. So it's important for the timing, the measurement of the movement. Let's review it one more time. Main points. The cerebellar cortex has 3 layers. That's different than the cerebral cortex, which has 6. There's a gray matter on the outside, white matter on the inside, and deep cerebellar nuclei in the middle. What I'm showing you right here is a cross-section where we're seeing the molecular layer, the Purkinje cell layer, the granule cell layer. The molecular layer has multiple fiber types. It has the dendrites of the Purkinje cells, and it has the axons of the climbing fibers and the granule cell parallel fibers. The granule cell layer, the Purkinje cell layer, has just one layer of cells, the Purkinje cells. The granule cell layers is very dense, looks like grains of sand. There's so many neurons here, so it kind of like can fill this up with granules of granule cell neurons. And these receive input from mossy fibers, whereas the climbing fibers go all the way up to the molecular layer. And that's the circuitry of the cerebellum.